Hello. Hi. We're learning about lysosomes. We're learning about cell biology and life science. Going through my notes. In a nutshell. So we're going through one organelle in the cell, which is the lysosomes, which is an important organelle. It is a membranous um, sac used to digest or degrade cellular macromolecules or even bacteria. It has a pH of 5, so it's very acidic. It contains hydrolytic enzymes used for breakdown of proteins, nucleic acids, lipids, glycolipids, glycoproteins, so various molecules. It has enzymes called hydrolases, which only function in the low pH and used for breaking down. Um, so, so various molecules are broken down that no longer are needed. After the material is broken down, lipids and amino acids are transported across the lysosomal membrane by permeases for reuse or like recycling and biosynthesis. Um, so, um, so sugars, proteins are broken down, various molecules, lipids, etc. So lysosomes contain up to 50 different degradative enzymes that hydrolyze the proteins, DNA, RNA, polysaccharides, lipids, etc. They receive content via vesicles. So the proteins used, the enzymes, enzyme proteins used in the lysosomes come from the endoplasmic reticulum, which is the organelle, which is continuous folds or sacs that contain ribosomes where proteins are produced. So the ER has vesicles that bud out that are sent to the lysosomes in order to do their degradative work. These enzymes and the environment of low pH is maintained by a hydrogen pump shown here. So this is a lysosome. So hydrolases are inside the protein enzymes. This is a hydrogen pump. So which uses ATP. The pumping requires expenditure of energy in the form of ATP hydrolysis since it maintains approximately 100-fold higher hydrogen concentration inside the lysosome than on the outside of this organelle. So outside the lysosome membrane in the cytosol, the pH is neutral, about 7.2, while inside it's about 5 due to these hydrogen pumps. So as ADP loses a phosphate groups becomes ADP, a hydrogen is entered into the organelle via the membrane channel. One of the major functions of lysosomes is digestion of material taken up from outside of the cell by endocytosis. Lysosomes are formed by fusion of transport vesicles budded from trans-Golgi network. The network, so the part of the Golgi which is on the away from the endoplasmic reticulum on the outside towards the cell membrane. So fusion of transport vesicles budded from trans-Golgi containing molecules taken up by endocytosis at the plasma membrane as well. So endosomes are coming from the membrane of the cell. They bring in molecules from the outside of the cell. Formation of lysosomes represents an intersection between secretory pathway through which lysosomal proteins are processed and the endocytic pathway through which extracellular materials are taken up at the cell surface. So these proteins come from the ER, the rough endoplasmic reticulum, which brings the enzyme proteins, and molecules taken up at the cell surface. So all these can be contents of the lysosome. Material from outside the cell is taken up in clathrin-coated endocytic vesicles, which bud from the plasma membrane, fusing with the early endosomes, which is part of the cytoskeleton linkage protein units at the membrane of the cell. Membrane components are recycled to the plasma membrane. The early endosomes mature into late endosomes, which are precursors to lysosomes. One of the important changes during endosome maturation is lowering of the internal pH to about 5.5, which plays a key role in delivering lysosomal acid hydrolases from trans-Golgi network. So the vesicles either come into the cell from the membrane or go out. So endocytosis or exocytosis, after the molecules are broken down, so protein fibers, subunits, known as endosomes, are used for this process. Hydrolases are in the acids inside the lysosomes that break down various molecules, sugars, acids, nucleic acids, carbohydrates, whatnot, whatever needs to be broken down. And transport vesicles help. So budding of vesicles. A molecule known as mannose-6 phosphate receptor is used for these vesicles. So from the Golgi, the budding towards the lysosomes. Molecules via the receptor vesicles bud, and the sugar phosphate receptor transport vesicles carry acid hydrolases, which are proteins that are bound for the lysosome, protein enzymes used for breakdown. Go to lysosomes. We'll continue in the next part. Thank you.